Hello, this lecture is going to cover Chapter 38, Biopharmaceuticals, which begins on textbook page 648. So the introduction there covers biopharmacology and defines it as the branch of pharmacology that studies the use of biologically engineered drugs. It then goes on to detail biopharmaceuticals as drugs derived from life forms that are existing in nature, such as plants and animals. So medications are produced by means other than direct extraction from a biological source when we talk about biopharmacology. So these things can be proteins like antibodies or even consist of DNA and RNA. So research is being conducted in this area, biopharmacology or biopharmaceuticals, to find new therapeutic medications for such life-threatening diseases as AIDS, various cancers, and even Parkinson's disease. So a majority of our biopharmaceuticals are derived from existing plants and animals. However, genetic engineering is another way to create these new drugs. So on textbook page 649, we're going to get into what are biopharmaceuticals. Well, biologics are medicinal products that are derived from blood, as well as vaccines, toxins, and allergen products. Our biopharmaceuticals are a class of therapeutic protein produced by biotechnological techniques, such as genetic engineering. So when we talk about biological medicine, we are talking about the use of biological systems, whether that be cells or tissues or biological molecules such as enzymes or antibodies in the manufacturing of commercial products, such as vaccines, blood products, allergenics, and proteins, including those antibodies. So to talk about this, we need a little bit basic understanding of recombinant therapeutic proteins, or RTPs. These are artificial forms of recombinant DNA that have been created by combining or inserting one or more DNA strands into a single molecule. Examples of products produced by this means would include humulan insulin, or RHI for recombinant human insulin, Cerezyme for Gaucher's disease, Leukine for neutropenia, and Synvisc or Hyalgan for rheumatoid arthritis. So recombinant DNA technology has four primary positive impacts on the production of pharmaceutically important proteins. One, it resolves source availability problems, so we don't have to worry about if these naturally occurring substances are readily available. We can engineer them, so to speak. It also addresses product safety problems because we can remove some of the disliked or unwanted effects, safety concerns, right? We can manipulate these things, so to speak. Three, it provides alternative to direct extraction from inappropriate or dangerous source materials. So the actual process of collecting some of these sources or materials from these sources can sometimes be dangerous to the person who's having to do the gathering. And so utilizing recombinant DNA technology, we are able to sort of sidestep or alleviate some of those safety concerns. And four, recombinant DNA technology aids in the generation of engineered therapeutic proteins, offering advantages over native protein products. So Table 38-1 details biologics that are made with DNA technology. This table is found on textbook page 650 in the top left corner, and you'll notice that most of the conditions listed here include things like rheumatoid arthritis, anemia from cancer, chronic renal failure, Crohn's disease, or even breast cancer and psoriasis. So let's look at how these drugs enter the market, how the FDA approval process goes, so to speak. And so this topic begins on textbook page 649, but it moves over to page 650 if you're following along. So the first phase we have here is the preclinical phase. This is when the new drug is tested against a targeted disease with the use of laboratory animals. And if things go well through this process, the company can then file an investigational new drug or IND application with the FDA. Now, this application needs to show the results of previous studies, that's that preclinical phase, and it needs to set out protocols for future clinical studies, how the company plans to progress forward in their studies. And they've got to be pretty specific. They should also include the chemical structure of the compound and how they plan to manufacture this for the masses. So this IND becomes effective if the FDA does not disapprove it within 30 days. So they don't necessarily issue an approval per se, but they can stop it if they see something that they don't like or that they think warrants further investigation before moving forward. And if none of those types of things cause the FDA to want to stop or slow the process, then they simply won't do anything. And after 30 days, that IND will then become effective. So future clinical trials would then include volunteers within clinic settings and hospitals under the care of a physician, and this step may take several years. Following the successful completion of all three phases, the company can then file a new drug application, or an NDA, if you will. And this part of the approval process can take up to 30 months. Now, think about that a little bit with regards to the COVID vaccination and some of the medications that have come onto the market as a result of the pandemic. If the approval process can take up to 30 months and this pandemic became an issue in the early part of 2019, and by the summer of 2019, we were already looking at vaccinations starting to come out onto the market, right? So that's a very fast track. So do know that, yes, the FDA did move the COVID vaccination process up. That was why most of those approvals were listed as emergency approvals, meaning that it had not fully completed this process of gaining FDA approval. It was on an emergent basis. Now, through the course of the pandemic, the FDA has since gone ahead and removed the emergent from a lot of these products, and they are now simply available on the market. But I want you to know and understand that the coronavirus has been around for a really, really long time. It's just that the version or the strain that came out in 2019 had been manipulated in a laboratory setting and then managed to escape containment. So a lot of those vaccines and drugs had already been kind of being studied for years, even decades, because the coronavirus is basically the underlying cause for what we typically refer to as the common cold. And again, I'm not trying to downplay the pandemic at all in any way, shape, fashion, or form, because again, that was a mutated strain that had been manipulated in a laboratory setting and then managed to escape containment. That was a big part of what made that version of the coronavirus so severe and escalated it very quickly into that pandemic status. So I just wanted to clarify that and address that a little bit while we were talking about the process of the FDA's approval of medications. So if you look at the bottom of textbook page 650, we have a figure 38-2 that details the approval process for a new biopharmaceutical product. I'm not going to necessarily read through this process for you, But I highly suggest that you do look through this, be very familiar with it. I can tell you that at least 2 or 3 of your test questions come out of this figure. So I'm going to go ahead and move on to the right-hand side of page 650, and we're going to begin talking about genetic engineering. So when we talk about genetic engineering, we are referring to the direct manipulation of an organism's genes to produce new medications. So there's 4 basic steps to this process. First, we're going to isolate the desirable genes, such as a resistance to a particular disease. Then we're going to insert that gene into a vector. Now, a vector is just any organism that does not itself cause disease, but can spread it by distributing pathogens from one host to another. Then we will use that vector to transform or genetically alter the cells of another organism. And lastly, that new genetically modified organism, or GMO, is isolated from the cells that did not take up the vector. And that GMO is then packaged with resistant genes and placed in a culture with penicillin to ensure that only the cells that have incorporated that vector actually survive. So on the right-hand side of page 651, we're going to get into stem cell research. So stem cells can renew themselves many times through cell division. These stem cells are unspecialized or undifferentiating, meaning that they do not perform specific functions within the body. Differentiation of stem cells means that they can transform into specialized cells, such as heart muscle, under certain circumstances. So, if we can reproduce something like heart muscle, of course, as you can imagine, this is very significant, right? Very strong advancement forward for medicine. However, when we talk about biopharmaceuticals, there is actually a political climate around this class of medications. And on page 652, we're going to look at that political climate. So again, the field of biotechnology is politically charged and highly conscientious. These medical breakthroughs are weighed against moral issues, such as, are scientists playing God by creating these new forms of life, right? We start looking into cloning and other things of that nature. We've made great strides in medicine as a result of these techniques, but at what cost? When we talk about stem cells, you need to know that human embryos are the source of stem cells. So again, are we taking a life to make these advancements? So at what cost are we striding forward in this field? And then, of course, there are concerns over health consequences for consuming genetically engineered plants and animals. The FDA has approved cloned meat onto the U.S. food market. So as you can imagine, that's a big concern for many people. So we're going to summarize chapter 38 at the bottom of page 652. Remember that your biopharmaceutical research is a growing field of pharmaceutics. There is a growing need for the drugs that are the end products of this research. Your diabetics need that insulin. That insulin comes about as a result of biopharmaceuticals. So, I mean, we're getting some really great medications out of this field. But again, for some of these advancements, at what cost have we reached them? Your biopharmaceuticals are used today to treat cancers, diabetes, hepatitis, multiple sclerosis, and other life-threatening conditions. So, I mean, it really is sort of a weigh the scales of justice because of the great things that this class of medications can do. But look what we're having to do to achieve them. So that genetic engineering and stem cell research also play an important role in discovery of new products and treatment of disease, but again, highly politically charged on benefits outweighing the risk, or, whatever bridges we've crossed that maybe we shouldn't have, if you want to word it that way. So I hope this has cleared up biopharmaceuticals for you. I do not necessarily care for the fact that we've had to do this through a recording. So I do want you to know that if you feel there is anything that is still unclear, that you didn't quite understand, you are free to reach out to me. I'm available to you. That's what I'm here for. Don't hesitate to let me know if you need a little additional assistance with this topic. I thank you for your attention, and I hope you have yourself a nice and great day. Enjoy your weekend.